It's Merger Wednesday. The countdown continues. 7 years until the convergence of biology and machine. Neural interfaces are no longer theory, they're the scaffolding of a new consciousness. But what does that mean for identity? For free will, for the soul? This is where we build the frameworks for ethical, philosophical, and spiritual growth for the next phase of human evolution. We're not just upgrading hardware. We're rewriting what it means to be human. Each week, we explore the biotech breakthroughs, decode the cognitive shifts, and prepare the mind for the merge. This isn't about fear, it's about readiness. Because the future isn't waiting. It's already wiring in. This is about preparing human consciousness for the most significant leap in our species' history. If you think this is just about chips and brains, you're not ready. But we'll get you there, one Wednesday at a time. So sit back and relax as we take you on a journey on Merger Wednesday.
Nice. Welcome. welcome to Merger Wednesday. Merger Wednesdays, people. Merger Wednesdays. And listen, I got a great show for you tonight. And this is going to be episode number 5 of Merger. Episode number 5. As we dive deep into this magnificent, highly advanced conversation. Look, I'm not going to hold the punches tonight. Tonight is going to be a difficult show in reference to the material, right? But I need you guys to follow along because this material is absolutely necessary, right? Absolutely necessary. So, if you haven't already, go get Go Time. Go get Go Time. All aboard the Fired Up train. This is your motivation manuscript done by yours truly with frameworks such as the 6 pillars of success, the cornerstone of magnificence, momentum code, discovering your why, all wrapped up in one right here. 330 some pages of motivation from a discipline aspect, right? Go get Go Time if you haven't already received it and gotten. If you get the hard copy in your local, I will come and sign I will come and sign your book, right? I will come and sign it. Oh man, let's do this. Let's get this started, man. Let's get this show started. And if you're new with us, if you're new here, look, thank you for joining us. Thank you for being here. You are a part of the Final Boss family, Final Boss Nation, Final Boss community, and we greatly appreciate you for joining us. And as always, go make sure you like, share, subscribe. Like, share, and subscribe all of the content, no matter where you see it, right? Go like and share it and subscribe it. Like, share, subscribe, people. Like, share, subscribe. I'm trying to change the ratio here. What do I want? I don't want that. I want, yeah, I like that one. Yeah, I like that one. So, yes, like, share, subscribe, people. Like, share, subscribe. And look, man, no matter where you see us, if you see us on Facebook, LinkedIn, YouTube, Twitch, Kick, or YouTube, and for your listening pleasure, make sure you check us out on Spotify. And if you haven't seen it yet, go to our website, man. Go check out our website and see all of the episodes, each and every last single one of the episodes as they are available to you on our website, man. Go check that out. And yeah, we appreciate that. Oh, by the way, all the comments, we greatly appreciate you for all of the comments that you guys are leaving, no matter where you're leaving it at. Thank you so much for that. And you can shoot us an email. You'd like to be on the show, you'd like to be part of the Final Boss podcast, Nation, Final Boss Network, Final Boss Community. Shoot us an email, man, so we can get that vetting process started. So you can be a part of the Final Boss team, right? The email is at info at one word, thefinalbosspodcast.com. Hey, Chakandi, how you doing? Hey, I'm telling you, this show is going to be heavy tonight. Make sure you take some notes. It's going to be heavy. Yeah, if you want to shoot, yes, let's get the vetting process started. Shoot us an email at info at thefinalbosspodcast.com so we can get that started. We're looking forward to having you part of the team and part of the Final Boss Nation. Yeah, man. And sorry for signing. I feel like I sound dry, but I'm not. I'm excited. I am super excited about tonight's show. And if you're just now joining us, thank you for being here. And guess what? Again, this is Merger Wednesdays, episode number 6. And tonight's title is we're going to be talking about the FDA-approved gene therapy. The FDA-approved gene therapy. Yes, thank you, Chagani. I just took them down. Thank you so much. Yeah, so tonight's title is the FDA-approved gene therapy. And it's about what you need to know, because a lot of you don't know what's out there. I guarantee it. So, man, if you're not familiar with MERGER, this is where human consciousness meets technological integration, right? Integration. So what's happening? What's happening is this. Tonight, we're going to be doing something a little different, right? Everything I'm about to tell you is authentic. All of our shows are authentic, but I like to say that on MERGER because we dive into topics that a lot of people may not deem is true until they go do their own research and find out that it is true. So I want to just give that disclaimer. Oh, and for entertainment purposes only, right? But tonight, especially, every fact, every number, every claim is all fact. This isn't speculation. This is documented reality that I'm using, right? I'm using this for my doctoral dissertation. And what does that mean to you, right? What does that mean to you? Because when I tell you the FDA has approved genetic modification on humans, I'm not giving you my opinion. I'm giving you facts you can verify yourself, and I'm giving you the sources to go check, right? Last week, we talked about CRISPR. Tonight, we're talking about what's already been approved, right? And what's already happening. What doctors are already doing to people's genetics with the blessing of our beloved government, right? This is research-backed, citation-supported, peer-reviewed reality, right? Now let's get into it. Man. But, we got to start with our brass tacks. And here it is, right? Here is the brass tacks. Let me get an evil laugh. Go ahead. Yeah. And here's the brass tacks. Understanding technological transformation requires distinguishing between documented reality and projected possibility. Gene therapy has transitioned from experimental to FDA-approved clinical intervention. The question for researchers is, what does this regulatory acceptance reveal about society's evolving relationship with genetic modification, right? And that right there comes from yours truly, Dr. AI. Right? So, let me Get some of my Java juice before we get started, man. For those who are just now joining us, thank you for joining us. Right, it's we're here we're here 5 days a week, 9 p.m. live. Catch us, right? Catch us, right? I want you guys to know a little bit about me. If you don't know, I'm a 2-time war veteran. I'm multiple business owner. I'm close to finishing my MBA at the University of Maryland, right? And I'll be starting my doctoral, right? I had to get that out there, right? Because right now I'm doing doctoral research. And I'm going to walk you through verified scientific evidence about tonight's topic. And I have to tell you this, right? On December 19th, 2017, a 7-year-old girl with a genetic mutation called RPE65 deficiency, right? Which was making her go blind. She underwent a surgical procedure. Doctors injected a virus carrying a corrected gene directly into her retina, right? 3 months later, she could see. This isn't a story I made up, people. This is published in The Lancet, one of the world's top medical journals. Russell and colleagues published the phase 3 clinical trial results, and 93% of patients showed dramatic improvement. 27 out of 29 people, the benefits lasted through 4 years of follow-up. The treatment is called Luxturna. Luxturna, right? It's FDA approved, and it's been approved since December 19th of 2017. It's the first gene therapy for an inherited disease ever approved in the US, right? Now, here's why I've been so specific about that, right? And especially with the sources, right? This episode is part of my doctoral dissertation at CTU. I'm researching AI foundations and AI ethical considerations, right? And that sounds like a fancy academic speak, right? For how do we maintain control over ourselves when technology starts merging with our biology, right? And that includes gene therapy. And this is where the merger starts. This is the conversation we're having. So everything I tell you tonight has a source. It's going to have a citation. I've done extensive research, right? A way for you to verify yourself, right? Because smarts don't just believe things, they verify. And by the end of our show, you're going to know, number one, what gene therapies are FDA approved right now, this instant, on January 21st, 2026, right? Right now. With the actual approval dates and published clinical data. Number two, how the FDA decides what genetic modifications are acceptable based on documented regulatory pathways, right? And number three, why this matters for understanding where we're headed based on analysis of current evidence rather than speculation, right? So let's get into it, right? Let's get right into it. But before we go any further, let's make sure we're all speaking the same language, because precision matters, right? I like to speak as if I'm a sniper, right? Because when you're doing research, You need to be precise. And the FDA defines gene therapy as this right here. Gene therapy seeks to modify or manipulate the expression of a gene or to alter the biological properties of living cells for therapeutic use. And here's what that actually means in plain English, right? I'm going to break it down for you. Your DNA is basically an instruction manual for building and running your body. Every cell has a copy. Those instructions tell your cells how to make proteins, and proteins do everything. Proteins build your muscles, proteins digest your food, proteins fight infections, proteins fire your neurons, right? So you think. Now, imagine one of those instructions has a mistake, right? A small mistake, a typo even, right? And instead of add 2 cups of flour, it says add 2 cups of flooring, right? You follow where I'm going with that, right? You follow the recipe. You're going to have some issues. That's what genetic diseases are, errors in the instructional manual. Now, gene therapy is one of those things, right? The gene addition is if you don't fix error, you just add a correct copy alongside it. Your body uses the good instructions and ignores the bad ones, right? And gene editing. This is when you use tools like CRISPR to go in and actually fix the error, cut out the mistake, replace it with the right sequence, okay? And gene regulation. You don't change the instruction. You just tell your body to use them more or less, like putting a volume knob on a specific gene. You turn it up or you turn it down, right? Now, this piece right here, I want you to listen to very carefully. Now, look, don't you go nowhere. We just getting started. Stay right there, right? Stay right there, right? Now, the FDA makes a huge distinction between 2 types, right? The first type is somatic gene therapy, which means you're changing cells in your body, your blood, your muscles, your eyes. These changes affect you. They don't get passed on to your kids. This is what's currently approved, right? Then you have germline gene therapy, and that means you're changing eggs, sperm, or embryos. These changes affect your children, your grandchildren, every descendant you'll ever have. The FDA would not approve that, period, right? They won't even review applications for it. That's documented policy. That is not my opinion, right? And what does this distinction matter? Why does it matter? Why? Because somatic therapy means modifying one person. Germline means modifying the human species itself. The FDA has said yes to the first. They've said absolutely not, hell no to the second. For now, right? For now. And understanding where that line is and whether it holds, that's a part of my research, right? That's a part of my research. So, how does something go from wild science experiment to FDA approved treatment? You can FDA treatment that you can get at the hospital, right? Let me walk you through the actual process, because understanding this tells you how carefully or not our beloved Gov is regulating genetic modification, right? Because it starts in a laboratory. Scientists testing on cells in dishes, then mice, then primates. This phase typically takes 3 to 6 years. Most ideas fail here. They don't work or they're too dangerous, right? But if it works, they file an investigational new drug application with the FDA. The FDA has 30 days to say yay or nay, right? And if the FDA says yay, clinical trials begin. And those trials have 3 phases, right? And we're going to talk about it. The first phase, phase 1. Phase 1 is 20 to 80 people. The only question is, will this kill you? Sounds harsh, but that's the reality. They're testing safety, not whether it works, right? And phase 2, 100 to 300 people. Now they're asking, does this actually do what we think it does, right? And then phase 3, 1,000 to 3,000 people. This is confirmation. This is where proving it works consistently across different populations, right? All 3 phases combined, 6 to 10 years. Then they submit a biologics license application. All the data, manufacturing details, safety profiles, right? The FDA reviews for 6 to 12 months. And then guess what? You add it all up, right? By the time you add it all up, it's 15 to 20 years from initial conception to FDA approval. $1 to $2 billion in development costs, right? And this is where it gets really interesting, right? Hola, it gets really interesting. But here's what I want you to catch. And this is for my warriors. That timeline is shrinking. The FDA created something called regenerative medicine, advanced therapy designation in 2017. It's an expedited pathway. They're speeding things up, right? Why? Because they see where genetic medicine is going and they're trying to keep pace with the technology. And once things get approved, it's available within months. Lux Turner was approved December 2017. By March 2018, kids across America were getting their vision back, right? That's the bridge we're on, from impossible to improved. And the bridge is getting shorter every single year. That's not speculation. That is an observable pattern that is in the data. Go check it out. Now, let's talk about Lux Turner, right? Let's talk about it. December 19, 2017. Mark that date, right? This is for your research. That's when FDA approved Lux Turner, the first genetic gene therapy for an inherited disease. And it wasn't just another drug approval. It was the moment genetic modification of humans became officially sanctioned medicine in our beloved country. Now, let me tell you, Lux Turner, what it actually does. Let me tell you about it, what it actually does. It's real science here, people. It's real science. There's a gene in your retina called RPE65. It makes an enzyme that's essential for vision. If both copies of that gene are broken, one from mom and one from dad, you develop inherited retinal dystrophy, right? You're born with vision, but it deteriorates slowly, progressively, until the point you go blind. No cure, no treatment, just darkness. But now with Lux Turner, it has the mechanisms scientists have been looking for, right? What it does is takes a working copy of RPE65 gene and package it. And they package it inside of an adeno-associated virus, specifically serotype 2, AAV2. They're engineered the virus so it can't replicate, can't make you sick. It just delivers the vehicle, right? They inject that virus directly into your retina during surgery. The virus does what the virus does. It gets inside your cells, but instead of making you sick, it delivers the working gene. Your retina cells start producing the enzymes you were missing. And eventually, you can see again, right? Russell and colleagues published the phase 3 trial in the Lancet in 2017, a multi-luminescent mobility testing. That's the gold standard for measuring functional vision. 93% of treated participants showed improvement at 1 year. 27 of 29 people, and the benefits lasted even longer. After a 4-year follow-up, data showed sustained improvement, right? Sustained improvement. Kids who couldn't navigate their own homes, running, playing with friends, reading, and adults who'd been functionally blind for years, we're seeing again. And get this, the cost, right? The cost of that. A whopping $425,000 per eye. Total cost for both eyes, $850,000, right? Imagine that. Now, I know some of you are thinking, that's insane. Who can afford that? And you're right to ask. We'll get to the economics later because it's just a major issue in the research. But right now, I want you to understand why Luxturna matters beyond saving people's vision. It proved 3 things that changed the game. Number 1, gene therapy works in humans, not in mice, not in theory, in actual human beings with published results, right? Number 2, the FDA will approve permanent genetic modification. This is an pill you take daily. This is one treatment that changes your genetics forever. And number 3, viral vectors can successfully deliver genetic material to human tissues. The delivery system works, which means it can be adapted for other genes, other tissues, other purposes, right? And Luxturna was proof of concept, an entire field of medicine. And once you prove the concept, everything accelerates. Ladies and gentlemen, motivators, smarts, warriors, final bosses. That's not my opinion. That's data, right? That's data. Now, let's pivot for a second. On May 24th, 2019, the FDA approved a therapy called Zolgensma. Zolgensma. That's Zolgensma. Zolgensma, right? The price for this therapy, get this, is a whopping $2.125 million. $2.25 million. It's a single treatment. It's the most expensive medicine ever approved in history at that time. And that was in 2019. And here's the thing. The data suggests it's worth it. Now, let me tell you about spinal spinal muscular. I don't know what I was going to say there. Spinal muscular atrophy, or SMA. It's caused by mutations in a gene called SMN1. That gene tells your body how to make the protein that keeps your motor neurons alive, the nerve cells that control your muscles. Without that protein, those neurons die. And when they die, muscles stop working. Babies with severe SMA type 1 can't sit up, can't hold up their heads, can't swallow properly, can't breathe on their own. Before Zolgensma, the published data showed the median survival age of 10 to 16 months. Most passed before they were 2 years old. Zolgensma delivers a working copy of the SMN1 gene using a viral vector called AAV9. This is important because AAV9 can cross the brain barrier. We were just talking about blood-brain barrier, right? Which most things can't do. 1 IV infusion, 1 time, 1 hour, $2.25 million, right? Now, here's the research. Mendel and colleagues published the results in the New England Journal of Medicine in 2017, before it was approved. But these were the pivotal trials. They enrolled candidates with SMA type 1, primary endpoint, survival at 20 months of age. The results were significant survival advantages, right, compared to historical controls. Babies who would have passed were living, and not just surviving, achieving motor milestones, sitting, rolling, some even walking. The 5-year follow-up data came out of 2022, and the Lancet Neurology sustained benefit. These aren't temporary improvements. This is permanent change to how the bodies function, right? So, with that being said, that price tag, $2.25 million, right? 2.25. The Institute for Clinical and Economic Review, the ICER, that's an independent nonprofit that does cost effectiveness research. They published an analysis in 2019. They compared the one-time Zolgensma cost to lifetime SMA treatment. Before gene therapy, managing SMA meant lifelong medications, constant medical care, assistant devices, lifetime costs. Those costs, $4 to $5 million. So mathematically, Zolgensma saves money over a patient's lifetime. But get this, and this is big, right? This is big. That only works if you can pay the upfront costs. Insurance companies struggle with writing a $2 million check, even if it saves money long-term. Research by Belter and colleagues in 2020 documented access disparities. Not everyone who medically qualifies for treatment receives it. Insurance coverage varies. Prior authorization is complex. Geographic access is unequal, which raises a question that's central to my dissertation research. If genetic modification technologies provide significant health benefits, but economic barriers prevent equal access, what does that mean for human anatomy and sovereignty? Because right now, whether you can get your genetics modified depends partially on whether you can afford it or navigate insurance bureaucracy. That's documented reality, not speculation, right? Now, we're going to pivot again. Let's go back in time to August 30th, 2017. Same year, as Luxturna, right? The FDA approved something that honestly sounds like science fiction, right? And that is called CAR, C-A-R-T cell therapy, CAR T cell therapy, right? And let me explain what this actually is because the mechanism is remarkable. Your immune system has these cells called T cells. They patrol your body looking for threats, bacteria, viruses, infected cells, right? When they find something dangerous, they attack and destroy it. But cancer cells, they're your own cells gone rogue. Your T cells don't recognize them as threats. They just ignore them while the cancer grows and spreads until you're unalive, right? CAR T therapy genetically modifies your T cells to recognize and kill, get this, kill the cancer cells. And here's the actual process, right? Of course, it has a process, and I'm going to tell you about it. Step one, right? Step one, leukapheresis, right? They extract your T cells through a machine that filters them out of your blood, right? And step two is the laboratory genetic modification. They use a lentiviral or retroviral vector to insert new genetic instructions, a chimeric antigen receptor. That's what CAR stands for. This receptor teaches your T cells to recognize specific proteins or cancer cells. And step three, the cell expansion. They grow those modified cells from millions to billions, creating an army of genetically engineered immune cells. And step four is the infusion. They put them back in your body. Those cells circulate, find the cancer cells, and destroy them, right? The FDA has now approved six, get it, six CAR T therapies. And I'm going to list them because this is verifiable, right? In 2017, the first one was called Kymriah, right? Kymriah. The second one was called Yescarta. The third, well, let me give you the date. Let me give you the years. I apologize. Let me backtrack, right? Kymriah was 2017. Yescarta was also 2017. Tecartus was 2020. Breyanzi was 2021. Absema was 2021. And Carvic was 2022. All right. Nelapu and colleagues published a pivotal trial results in the New England Journal of Medicine in 2017. Complete response rates, that means cancer completely gone, ranging from 40 to 80%, depending on the specific cancer type and how many prior treatments the patients had, right? Locke and colleagues published a 5-year follow-up data in the Lancet Oncology in 2019. Durable responses, people with terminal cancer given months to still be with us, cancer-free years later. But let me be clear about the risk because academic integrity requires honesty, right? Maud and colleagues characterized adverse events in NEGM in 2019, right? Cytokine release syndrome occurred in 77% of patients. That's where your immune system goes into overdrive. Fever spikes from 105 to 106, right? Blood pressure would crash, organs would fail. It can be fatal, right? Management protocols exist now, right? Management protocols exist. But CAR T therapy is dangerous. You're talking about a treatment where you genetically are modifying cells that might cure your cancer or might trigger a cascade that kills you faster than the cancer would have, right? And yet thousands of people choose it. Because when you're staring at the D word itself with no other options, the risk benefit calculation changes dramatically, right? That's going to be relevant later when we talk about using gene therapy for non-life threatening purposes. Because the risk tolerance question becomes very different when you're not being unalived, right? Now, Final Boss community, Final Boss nation, Final Bosses, listen up. This is the date everything changed. And that date was December 8th, 2023. And that's when the FDA approved Casgevy, right? Casgevy for sickle cell, right? And Casgevy uses CRISPR, the first CRISPR gene editing therapy ever approved for human use. And last week, I told you about CRISPR. I told you about it, right? Tonight, I'm documenting it, right? So let me give you the background. Sickle cell disease affects approximately 100,000 Americans. It's caused by a single point mutation in the beta globin gene, right? One letter in the genetic code gone wrong. That mutation makes red blood cells rigid and crescent shaped instead of round and flexible. Those rigid cells get stuck in blood vessels, block blood flow, cause excruciating pain crisis, damage organs, shorten lifespans. It's brutal. But for decades, we couldn't fix it. Pain management, blood transfusions, uh, hydroxyurea was used to reduce symptoms, but no cure. Right? No cure. But here's how Casgevy works. Doctors extract your hemotopic stem cells, right? The cells that make blood cells. They take them to a lab. They use CRISPR-Cas9 to edit a gene called BCL11A. They're not fixing the sickle cell mutation directly. Instead, they're reactivating a gene that makes fetal hemoglobin, right? Babies don't get sickle cell symptoms because they have fetal hemoglobin. After birth, your body switches to adult hemoglobin. And that's when the mutation causes problems. Casgevy switches it back. Then you undergo myeloablative conditioning, right? That's chemotherapy to destroy your existing bone marrow. They infuse your edited stem cells back. Those cells repopulate your bone marrow, start making blood with fetal hemoglobin, and the sickle cell crisis stops, right? Franguel and colleagues published the initial trial results in NEGM in 2021. They enrolled patients with severe sickle cell disease. Primary endpoint, elimination of vaso-occlusive crisis. Those painful episodes, right? Very, very painful. The results, get this, 28 out of 31 patients, 28 out of 31 patients, that's 90%, were free from severe crisis for at least 12 months. Median follow-up was 19.3 months, and they sustained elevation of fetal hemoglobin, and it was documented. The FDA also approved a second therapy the same day, right? Lifegenia, which uses a different approach, gene addition with lentiviral vector rather than CRISPR editing. Cost data, Casgevy list at 2.2 million. Lifegenia at 3.1 million, right? This is CRISPR editing human genes, FDA approved, and it's happening right now. This isn't sci-fi. This is sci now. The technology to precisely edit genetic sequences is no longer experimental. It's clinically available. The regulatory precedent is set. And here's what I want you to understand about CRISPR, right? CRISPR doesn't distinguish between fixing a disease mutation and modifying any other genetic sequence. The tool works the same way, regardless of the application. The FDA approved the tool for therapeutic use. That's the current regulatory boundary. Whether that boundary holds as the technology advances, that's an empirical question requiring continued observation, not a prediction that yours truly can make, right? But the tool is approved. That's documented fact, right? Now, we talked about what's already approved. Now, let's look at what's currently in the FDA pipeline, because this tells us about research trajectories without speculating about outcomes, right? And according to the FDA public database and the Alliance for Regenerative Medicine 2023 report, there are currently over 200 gene therapies in active clinical trials. But let me break that down, right? Let me break that down in what's in late stage development, right? And we're talking about phase 3 trials. And I talked about phase 3 trials in the beginning. So listen up. These phase 3 trials, which are closest to potential approval, right? Whoa. What was that? Give me a second. One of my cameras failed. Imagine that. Stay there. All right. Phase 3, right? Clinical trials. You can check this out on clinicaltrials.gov, right? And it shows several active trials targeting dystrophin gene mutations. I'm not predicting when they'll be approved. I am documenting that they're in the advanced testing stage, right? Hemophilia, right? This is two therapies where these two therapies were actually approved recently, right? Roctavian for hemophilia, and that was in June of 2023. Then you have Hemgenix for hemophilia B. That was in November of 2022. Hemgenix is currently the most expensive drug ever approved at $3.5 million. That's how much it costs to get the gene therapy for Hemgenix, right? Now, Thompson and colleagues published gene therapy results in NEJM in 2018. Multiple candidates in late-stage trials using similar approaches to the sickle cell therapies, right? Now, phase 2 trials, we just talked about phase 3. This is what's in phase 2, right? Age-related macular degenerative or degeneration, right? This is there are several gene therapies approaches for vision preservation, right? Campochiro and other colleagues published early results in Human Gene Therapy 2017, right? And then there's something that's out there that's called Huntington's disease, right? This is still phase 2. This is gene slicing approaches using antisense oligonucleotides, right? And Tabrizi, who's Dr. Tabrizi, and colleagues published in phase 1 and phase 2 results in NEJM in 2019. And here's what's in development in the early stages, right? In medicine for Alzheimer's disease. Multiple preclinical and phase 1 approaches exist, but these are in the very early stages, right? I'm telling you, I'm documenting they exist, not claiming they're close to approval. Then you have age-related research. This is predominantly preclinical, Baker and colleagues published research on syncytin cell clearance in Nature in 2016. But clinical applicants or clinical applications remain distant, right? Now, let me be clear. Let me be clear. And let me be very, very clear about this, right? That pipeline of data that I just gave you, right? Just because something's in clinical trials doesn't mean it will get approved. Historical data from Hay and colleagues shows only 12% of phase 1 therapies eventually reach approval. The average time from phase 1 to approval is 10.5 years, right? So when I tell you 200 plus therapies are in trials, I'm not predicting 200 new trials. I'm documenting current research directions, right? And what does that tell us? What does that tell us? It tells us that the research is expanding from single gene rare diseases toward more complex conditions. That's observable in the pipeline data, where those translate to approvals and when remains to be seen. That's the academically honest way to present this information, right? Now, man, let's talk about what can go wrong. Let's talk about what can go wrong, right? Because it's important. This is important, right? I have to be honest. This is part of my dissertation piece, right? Academic integrity means I can't just tell you about the successes. I have to tell you about the failures too. Because gene therapy can fail, and sometimes it fails catastrophically, right? Catastrophically. Without mentioning any names from the research that I've done, there was a gentleman who was 18 years old. He had a genetic liver disorder, right? Ornithine transcarbamylase deficiency, but it was manageable with diet and medication, not immediately life-threatening. He volunteered for a gene therapy clinical trial at the University of Pennsylvania. He wanted to help advance science. 4 days after treatment, he was no longer with us, right? The adenoviral vector triggered a massive immune response. It caused a cytokine storm, which is multiple organ failure, right? And there was nothing they could do to save him, right? Raper and colleagues published the analysis in molecular genetics and metabolism 2023. This passing almost destroyed the entire field of gene therapy. The FDA imposed strict oversight. Trials were suspended, funding evaporated. It took years to recover, right? And then the next case, right? The next case, there was a SCID trial. SCID trial, right? In the early 2000s, right? They call it bubble boy disease. And kids born without functioning immune systems. The gene therapy worked. These kids developed immune function. It was successful until several of them developed leukemia years later, right? Hansen, Bay, and Abena and colleagues published the analysis in Science in 2003. The viral vector had inserted the therapeutic gene right next to the cancer-causing gene called LMO2, and it was activated, which caused leukemia. These aren't rare flukes. These are the inherent risk of genetic modification, right? Now, there's more, right? Calcido and colleagues published research in the Journal of Infectious Diseases showing 30 to 60% of the population has pre-existing antibodies to AAV vectors that can reduce efficacy of increased adverse reactions. CAR T therapy causes cytokine release syndrome in 77% of patients, according to Lee and colleagues' research in Blood, right? That was the magazine Blood, right? Insertional mutagenesis, the risk of genes insert in the wrong place and could cause cancer. Baum and colleagues reviewed this information in Blood in 2003, right? And then there's off-target effects. CRISPR can cut unintended gene sequences. Zhang and colleagues published analysis of this in Molecular Therapy Nucleic Acids in 2015, right? And then there's long-term unknowns. Most approved therapies have less than 10 years of follow-up. The FDA requires 15-year monitoring, but we don't know what happens at 20, 30, or 40 years, right? The FDA response: risk management. They implemented risk management. They created risk evaluation and mitigation strategies, right? For specific therapies, or REMS. Mandatory long-term follow-up studies, patient registries for surveillance, and adverse event reporting through FAERS database, right? Now, here's what the evidence shows, right? We're modifying human genetics with less than complete understanding of long-term consequences. That's not fear-mongering, people. That's factual observation of the timeline. Most therapies were approved with 5 years or less of follow-up data. For life-threatening diseases like terminal cancer or fatal genetic disorders, the FDA has determined the benefits outweigh the risk. That's evidence-based regulatory decision-making. But the question of risk tolerance changes when you're talking about non-life threatening applications, right? That's the area requiring continued research and policy analysis, right? Because the FDA approves gene therapy for treating disease. That's the current regulatory standard. But here's where it gets complicated. And there's extensive bioethics literature on this particular issue. What exactly counts as disease? That's the question. What's treatment versus enhancement, right? Ju Ingus published a foundational analysis in the Journal of Medical Philosophy in 1997. Can enhancement be distinguished from prevention in genetic medicine? He identified the definitional challenges we're still grappling with today. And this is something we're still grappling with today, right? Now, let me give you some examples that bioethics scholars debate, right? The first is if a child has growth hormone deficiency and would only reach, let's say, 410, treating that seems reasonable. But what if they're 5'6", little short, but normal, and parents want gene therapy to make them 6'2"? The question is, where's the line? Where do we draw the line, right? And then there's cognitive functions. If someone has an intellectual disability IQ of 65, intervention seems like treatment. But what if somebody has an IQ of 100 and wants gene therapy to boost it to 130? Or like me, who has an IQ of 130, and I decide I want to enhance that. I'm just joking. But look at this. Listen, look at this. If you're legally blind, correction is clear treatment. But if you have a mild myopia, what if you want better than normal vision? What if you want eagle vision? Where does treatment end and enhancement begins? This is important, right? This right here is very, very important. The current regulatory framework says the FDA evaluates therapies based on diagnosed medical conditions, but medical diagnoses evolve threshold judgments, right? Below this line equal disorder. Above this line equal normal variation, right? Cohen published analysis and Tulsa Law Review in 2013 examining what, if anything, is wrong with human. What is anything wrong is wrong with human enhancement? The literature shows this is a philosophy that's very complex, right? Very complex. Multiple scholars have identified enhancement challenges, seeking treatment in jurisdictions with different standards, right? Using approved therapies for non-approved purposes, right? And then operating in regulatory gray areas. These aren't predictions. These are documented concerns in the academic literature, right? And listen, I'm very clear again. I am not claiming this, right? Now, I want you to understand something, and neither am I predicting, right? Therapy enhancement boundaries will eventually disappear. I'm documenting that. The boundary exists in current FDA policy. Bioethics literature identifies definitional challenges, right? Enforcement complexities have been documented. International regulatory variation does exist. Whether and how the boundary evolves is an empirical question requiring ongoing observation, not something that I can predict. But understand, the current regulatory landscape and scholarly database, and those who debate this, debate that it is essential for research in this area, right? Now, I'm going to connect the MERGER framework, right? We're going to talk about the MERGER framework layer 1. And I'm definitely going over probably about 20 minutes. So stay with me. Stay with me, people. Let's connect this to the broader research framework. My dissertation examines technologies potentially relevant to human AI integration architectures. Gene therapy represents one component I'm analyzing. The theoretical framework I'm developing proposes a 3-layer architecture, right? Layer 1 is the biological foundation. That's the neural substrate characteristics, cellular level system, genetic architecture, and gene therapy that operates at that layer. Layer 2 is the interface technology. It's the brain-computer interfaces, neural recording systems, signal processing. And that's our next episode of Focus next week, episode 7, right? Of MERGER, right? And then there's layer 3, the integration and architecture. That's the consciousness interfacing, information exchange frameworks, hybrid cognitive skills, and theoretical exploration, right? Now, published research suggests ways that genetic modifications can theoretically support neural interface integration. Listen to me very carefully. I'm going to be very clear to cite these sources, right? Because biocompatibility enhancement is research right now, research on reducing immune response to implanted devices that exist. Rosenberg and Warobach published analysis of immunogenicity in biodrugs in 2004. And their question was whether gene therapy could enhance device biocapabilities remains theoretical, right? Neurofunctional optimizations, right? Bauwieler and colleagues published research on brain plasticity in Annual Review of Neuroscience in 2012. Neuroplasticity is relevant to interface adaptation, but whether gene therapy could enhance it is speculative, right? You heard that, right? And the next one, longevity considerations. Extended health spans could theoretically support long-term integration. Research on cellular synthesis exists. Baker and et al. In Nature 2016. But aging interventions remain largely preclinical, right? And let me be clear. If it's speculative, I'm going to let you know it's speculative or there's speculation. These remain theoretical applications, not gene therapies, right? No gene therapies have been approved for neural interface preparation or enhanced purposes. This is analytical framework development, not prediction, right? The research asks a question. The question is, does regulatory acceptance of somatic genetic modification for therapeutic purposes establish precedent that informs future discussions about genetic modifications supporting technological integration? That's the question I'm examining, not answering, examining. But look, here's what's documented. FDA has approved permanent genetic modifications for therapeutic purposes, right? Take note of that. Published research explores various potential applications of genetic modification. Take note of that. Bioethics literature examines boundaries between therapeutic and other applications. Take note of that. How these elements interact as technologies advance is an empirical question requiring continued observation. That's the academically responsible way to frame this very analysis, right? Right. Now, everything I've discussed tonight has been about the FDA, American regulations, but we need to understand international context because gene therapy development is global, right? In 2018, Chinese researcher He Wankui used CRISPR to edit CCR5, the CCR5 gene in human embryos. Twin girls were born with modified genomes. The international scientific community condemned him, and Chinese authorities imprisoned him for years. Cyranovsky published detailed reporting in Nature 2019. Why? Right? Why this matters? This case demonstrates technical feasibility of germline editing. He did it. Regulatory variation across jurisdictions. It was in China. International consensus against current germline modification was broad condemnation, right? Enforcement exists but varies. Yeah, he was punished, right? China subsequently strengthened regulations. The Xinhua news agency reported new ethics rules in 2019. Updated regulations were established and published in 2023 by China's National Administration of Disease Prevention and Control, right? And based on published regulatory analysis, our beloved country said this: somatic therapy is an FDA-approved pathway. Germline modification explicitly prohibited. Funding restrictions through congressional appropriations riders is no good. right? The European Union had an answer. Genetically similar framework to us. European Medicine Agency coordinates, right? Some variation by member state. But in China, post Dr. He, post, the oversight was strengthened. Clinical trial registration was required. Less transparency or less transparent public disclosure, right? And in other jurisdictions, significant variations in regulatory capacity. Some countries lack specific gene therapy frameworks. Some create potential for medical terrorism, right? Understanding regulatory heterogeneity is essential for analyzing how technologies diffuse globally, what governance challenges emerge from jurisdictional differences, how international variation affects research trajectories. This doesn't tell us what will happen. It documents the current landscape that research must consider, right? And I'm telling you this from an academic note. I'm documenting regulatory variation without predicting future policy changes or speculating about international competitive dynamics. Those would require different methodological approaches beyond current evidence synthesis, right? Now, to my final bosses, to my warriors, to my smarts, to my motivators, right? To the final boss community. Let's synthesize what we've documented tonight based on variable evidence, based on documented fact. FDA has approved multiple somatic gene therapies from 2017 to present. There are approved pathways, right? Approval of those pathways is established and being actively used. Over 200 therapies currently in clinical trials. Regulatory framework includes expedited pathways, right? And number two, the clinical efficacy, right? This is the published data. And phase 3 trials demonstrate significant benefit for approved indicators. Long-term follow-up, 4 to 5 years, shows sustained effects for several therapies. Results published in peer-reviewed journals, right? All that have been cited tonight, right? And the safety profile. This is all evidence-based and is documented. And we've documented adverse events that range from manageable to severe. Historical failures influence current protocols. Long-term effects beyond 10 years remained under investigation. Risk-benefit assessment varies by disease severity. And we talked about cost. The costs range from $373,000 to $3.5 million, right? We talked about the cost effectiveness that demonstrated over long horizons in some analysis. Access barriers documented in published research. Payment models that are currently evolving, right? And then we need to discuss the regulatory boundaries and do a policy analysis, right? Because current approvals are limited to therapeutic applications. Bioethics literature identifies definitional challenges. International regulatory variation creates governance complexity, right? Research questions emerging from this evidence, right? Here's the question. Number one, how does regulatory acceptance of permanent genetic modification influence societal discourse about genetic intervention boundaries? And number two, what mechanisms could ensure equitable access when economic barriers exist? And number three, how do regulatory boundaries between therapy and other applications function as technological capabilities advance? And number 4, what role does international regulatory variation play in shaping global practices? And number 5, how does somatic gene therapy precedent inform discussions about other forms of human biological modification? Now, I want you guys to understand, right? I know this has been a night that's been full of word salad, right? Word salad. But I have to let you know it's for a purpose. So stay with me, right? Stay with me. I'm dissertation positioning, right? I'm doing a dissertation position for my doctoral research in AI foundations and AI ethical considerations, right? Right. Gene therapy represents this: a case study in regulatory negotiation of human modification, right? That's been documented. An example of technology transitioning from theoretical to approved. That's verifiable. A precedent potentially relevant to the future. Biological technological integration discussions, right? That's analytical framework. And a lens for examining how society determines acceptable modification boundaries, right? That's research focused. What I'm not claiming, and hear me loud and clear, I am not claiming this. I am not predicting specific future approvals. I'm not claiming enhancement applications are inevitable. I'm not making deterministic statements about technological trajectories, right? But what I am documenting is this: current regulatory status, published clinical evidence, documented economic barriers, identified policy challenges, and research questions requiring continued examination. That's the difference between rigorous research and speculation, right? We're almost at the end. Like I said, 20 minutes over. Man, that was accurate, man. That was accurate, right? We've covered a lot tonight: warriors, motivators, final bosses, and smarts. And let me bring this home. What we documented with citations were multiple FDA-approved gene therapies from 2017 to the present. We established regulatory pathways with ongoing evolution. We've talked about published clinical evidence showing efficacy for specific identifications, right? We documented safety considerations requiring long-term monitoring. We discussed economic and access challenges backed by research. Information was conveyed about regulatory boundary questions that identified in scholarly literature, right? And finally, international variation in gene therapy governance. Every claim I made tonight has a source, a citation, a way for you to verify it, because this is doctoral level research. And doctoral level research means you can check my work, right? And here's a methodology recap, right? This particular episode will distinguish between what has occurred, the FDA approvals, the clinical results, the regulatory decisions. What is occurring? Current pipeline, ongoing trials, policy discussion. What requires observation? Long-term effects, regulatory evolution, access patterns, right? It intentionally avoided the following. This is what we avoided, right? We avoided predictions about future approvals on timelines. We avoided speculative scenarios about technological trajectories. We avoided deterministic claims about inevitable outcomes. We avoided unverified predictions, right? That's how you have academic integrity in action, right? And for my dissertation, right? That research included longitudinal analysis of FDA approval patterns, comparative policy analysis over jurisdictions, economic modeling of access and equity, ethical framework development for modification boundaries, empirical studies of public perception, right? Now, let me drink some Java juice. We're almost done. Stay with me. That's good. Next Wednesday, July 28th, 9 p.m., right here on Merger. We're going to be talking about episode 7, right? Episode 7, we're going to be discussing the neural interface technologies, current FDA status, and clinical evidence, right? Same rigorous approach, same fact-based analysis. We're examining neural interface approvals, published clinical trials, current regulatory status, verified safety data. No speculation, all evidence, right? Now, look, I know this was dense. I apologize. I know that it's more academic than usual. I also apologize. I also apologize about the word salad that's throughout. That's necessary. I apologize about that. But there's a reason. Because when we're talking about genetic modification of humans, precision matters, right? I needed to deliver to you in a sniper effect, right? Because facts matter. Sources matter, because bad information in this area can lead to bad decisions, and bad decisions about genetic modification can have consequences. So yeah, I gave you citations, I gave you journal articles, I gave you FDA documentation. That's not to show off, man. There's no vanity here. This is all informative, and we're all on this journey together, building the Final Boss podcast community and the Final Boss Nation. That is to equip you, right? No, right? Because smarts don't just believe things, they verify things. And I gave you everything you need to verify in what I said tonight. Gene therapy has moved from theoretical to FDA-approved reality. That's documented. Understanding that the transition occurred provides essential context for examining broader questions about human biological modification. This is documented reality, not speculation. Published evidence, not prediction. And for my doctoral work and for this community, understanding what's actually happening is the foundation for meaningful analysis. I'll see you guys next Wednesday, right here at 9 p.m. for Merger Wednesday. Stay informed, stay critical, stay rigorous, man. Listen, this is me, the one and only, the Final Boss. We're going from chess piece to chess master, one evidence-based episode at a time. You guys have an amazing night. Thank you for being here. Thank you for allowing me to convey this information to you. Man, every fact cited is peer-reviewed source. Clear distinction between documented evidence, right? No speculative predictions. All conversational delivery that respects my audience that is of high intelligence, right? I thank you for being here. I appreciate you for being here. And if this resonated with you, let it marinate in your spine, right? And go share it with 3 other people, man. Share the information. Don't gatekeep. Share the information with 3 others, right? Have them come on board and join the Final Boss community, Final Boss Nation. And remember this before you go. There's a heavy pain that comes with discipline, but the pain of regret is even heavier. We are here. I am here. And as always, no matter where you're watching, good morning, good afternoon, good evening, and good night. And remember, always be balanced, be equal, and most importantly, be smart. That's our show for tonight, man. I greatly appreciate each and every last one of you. I can't thank you enough. Y'all have a good night. See you tomorrow for Special Guest Thursday, right? Special Guest Thursday, 9 p.m. tomorrow, right? Live. All shows are live. All shows are live. Y'all have a good one.